Good afternoon, everyone. I'm very excited to be here today to talk to you all a bit about Ensembl and how we've been scaling over the past few years for accessibility, performance, and interoperability. And I'm giving this presentation on behalf of the entire Ensembl team. I think many of you may be familiar with the Ensembl platform. It's both an Elixir core data resource as well as a global core biodata resource. You may also be familiar with our homepage shown here. The Ensembl platform hosts both analysis and interpretation of genome annotations. And so it integrates publicly available information across a variety of topics. So the screenshot here is just a particular region of a genome, and you can see there are tracks representing comparative genomics, different annotations for genes and transcript. Okay. This is slide right. Yes, but it's not showing up here. So okay. You may have seen this view of the Ensembl homepage. And here is a screenshot of a particular region of the genome. You can't really see on this small scale, but if you zoom in, you can see tracks representing comparative genomic analysis outputs, annotations across different genes and transcripts, functional annotations, so regions like enhancers and promoters, as well as tracks showing genetic variation information and regulatory annotations. And we both pull in a lot of these annotations and data from other public resources. I've listed a few here that are also EBI resources, including the ENA and UniProt. But we also perform our own analyses and display those results in the webpage as well. And so in this session on scalability, I thought I would sort of focus and give some examples of how Ensembl has addressed some scalability challenges recently. So Ensembl has been around for about 25 years and was initially developed to support the outputs of the Human Genome Sequencing Project, then expanded to support a few hundred different genomes spanning different key vertebrate species. And then most recently, with the advent of a lot of global biodiversity projects and pangenome projects, these have been producing thousands of genomes that Ensembl is also scaling to make sure that we can represent and use. And each of these released genomes contain full annotation and analysis sets, so all of those tracks that I mentioned on the previous slide. And you can see here just in the graph on the right, the increase in the number of species of genomes represented by Ensembl over the years, so clearly increasing and not really slowing down. So this presents a need to make sure that Ensembl can be scalable and also that we can rapidly deploy all these new genomes and the annotations so that they can be used immediately by the relevant communities. So with this challenge, Ensembl developed what we call a rapid release process. So we anticipate that we're actually going to expand to tens of thousands of genomes in the next few years. And so we needed to develop this rapid release process for genomes, especially that are of high interest to communities. So maybe it's the first genome released for a particular species of interest. So the way that we have done this is by reducing the number of annotations and associated information as part of these rapid releases. So it's really minimally the genome and some first pass gene annotations. And this enabled us to reduce the release time from about 2 to 3 months for a genome to about 2 to 3 weeks. And this, of course, depends on the scale or size of the genome. And you can see the plot on the right. These are the number of released species that we've been able to do since This process was released in 2020, scaling to a few thousand genomes. Another area that we've been able to scale is with our vertebrate genome annotation pipeline. So we've overhauled this in the past number of years. I think initially started in 2015, making the process more modular and fully automated. So we really only have human intervention in what was previously a manual process, only if something goes wrong. This process has benefited from having more species-specific curated protein sets available, as well as more transcriptomic data for single species. This has helped us improve the accuracy and speed of the annotations. And this was especially important because we realized that the volume of the number of genomes that we were annotating actually outpaced our ability to release them. So this tied in nicely with our development of the rapid release process. So I've explained these sort of two components of Ensembl, the sort of original OG Ensembl released in 2000, which actually contains a number of component sites if you've been on the site for plants and metazoa, for example. But this covers all kinds of species. We currently have over 33,000 genomes as part of the Ensembl resource. I mentioned we have these annotation pipelines that are in-house that we then produce results that are displayed in the browser alongside a lot of additional annotations. And we have the rapid release site, which was released in 2020, covering a fewer number of species, but still a really important resource for the communities. And these, as I mentioned, have sort of limited annotation in the first instance. With these two examples, I wanted to sort of think a bit broader in some of the ways that we think about how we approach different scaling solutions for sustainability. So one thing we think about is the value. So what is the value of the resources that we're currently providing? How are they impacting the community? How they're useful for those communities, and if there are other resources that could offer similar value as Ensembl. We, of course, also have to think about the cost to run and maintain both the services and the annotations that we're producing. So this is, personnel costs of compute and hardware, and we also do think about the environmental impact as well. So can we make our pipelines more efficient? Not just so that they're faster, but also to make sure we're limiting our impact on the environment. And then considering both the value and cost, we then can start to think and prioritize about what actions we would like to take. So where we might invest a bit more resources, how we could maybe scale our offerings, or in some cases reduce our offerings. And sometimes even thinking about what we might need to retire in order to scale efficiently. Oh, yep. So it's as Ensembl, it's our responsibility to make sure that we can, taking all of these into account, identify how we can best serve the needs of our community. So what can we take from lessons from this rapid release process and our genome annotation scaling to support scalability for the broader Ensembl? This is a diagram that we've produced on the current Ensembl release process. You are not meant to be able to read anything. That is intentional. But you can see there's lots of components, lots of places where something could go wrong, lots of dependencies. So it was really important for us to learn from that rapid release process and perhaps try to simplify our current process. So we have done this and have moved to a two-stage approach for releasing genome annotation. So we do a first, what we call a partial release. So that's just like that rapid release process where you have sort of the genome and some minimal annotations. And we will do this every 2 to 3 weeks. And then every about 2 to 3 months, we'll do a fully featured integrated release. So more similar to what we have now for the Ensembl releases. So this kind of gives you the best of both worlds. It makes some information immediately available, but also then follows up with the more integrated information. Just generally thinking about scalability, we think about how we can move to more modern infrastructures. So this includes things like databases, updating to the newest programming languages, using more modern technologies, and also making sure that we're following good software practices. Sometimes this does mean you need to re-implement things or restructure things. So taking into account how much effort that would be, and then the value we get from modernizing is something that we are thinking about all the time. And so I'll give one example with a standard that we've implemented. This is the RefGet standard, which is one of the Global Alliance for Genomics and Health, or G4GH standards. It's a way to reference sequences in a very efficient way by producing essentially what is a hash that you can use to look up a sequence of interest. This is a globally recognized standard with multiple implementations across the world. And we originally used the first reference implementation of RefGet in the Ensembl platform, but we realized that this didn't scale very well when we were addressing over 200 or over 2,000 genomes. And it was also not very stable, so it didn't really support our needs at the time. So what did we do? We re-implemented. So last year we created a new implementation of RefGet using more modern technologies. This one is written in Python. And we did some testing and discovered that for our system, it made sense to put effort into re-implementing something, re-implementing the RefGet standard. And so we saw much faster speeds to loading reference sequences, and it was much more reliable and stable. So this leads me to talk about our newest Ensembl platform, what we're calling now Ensembl Beta or Beta, depending on where you're from in the world. I will say Beta. The Beta Ensembl was released this year, but it's still running in concurrent with the main Ensembl site. But we did retire the rapid release because the Ensembl Beta now contains all of the rapid release annotations and data and processes. If you go to beta.ensembl.org, you can see the new homepage. I highly recommend that you check it out. We're also collecting feedback. So if you have any feedback about the site, please do share. The latest Ensembl Beta release with data was in, I believe, May of this year with over 3,000 genomes. That's when we retired the rapid release. It supports a large number of the current Ensembl processes and features. And we plan to launch it next year when it will be in full parity with the current Ensembl site. So if you're using current Ensembl, don't worry, it's not going away just yet. We did a lot of community research in both what features people wanted to see, what new data or annotations. And we're also developing now some really cool new features that will only be available in the beta site next year, including things like visualizations for structural variations, and also a new way to view regulatory annotations at a single cell level. So I've kind of given you just a few different examples of some of these broader topics that we are thinking about in Ensembl of how we can meet the scalability challenges we're facing. So I mentioned about streamlining our operations with our new hybrid release process, not being afraid to both adopt and adapt standards as needed. So it's great to use standards, global standards when they're useful, but not to be afraid to potentially re-implement if they serve your needs. And also just keeping an eye on ensuring that we have modern pipelines and infrastructure running, again, to have efficiency, but also be climate friendly. So with that, I'd like to thank the entire Ensembl team. There's a large number of people that it takes to maintain a resource like Ensembl. So if you've ever used Ensembl, maybe send a nice little message to our help deskers saying they're doing a great job. It's always good to hear positive feedback. I'd like to acknowledge our funders, and I'm happy to take any questions.
Thank you very much, Mallory. Are there any questions from the audience?
Yeah. All right, can you describe the push behind that, I would say the rapidly released genomes that are coming from and which outcomes?
Yeah, so caveat, this was before my time in Ensembl, but I believe that was initially a response to like COVID-19 coming. And so there were some genomes coming from there, but also that was at a point where we had a lot of the biodiversity projects coming out. And so there were just thousands of species, new species that we needed to handle. And those were close collaborations with those projects. So it wasn't like, an explosion of new mouse genomes, but it was insects and plants and these kinds of things.
Over here. Yeah, so tens of thousands of genomes in Ensembl. Genome annotation is never really finished. So how do you manage the continued annotation of everything that's in there while handling the new ones?
That's a good question. I will also caveat the annotation team is not my team. I believe they prioritize, right? So if there's a lot of new data coming out, for example, in the human pangenome projects with the references coming out, they will prioritize human annotations because there's a lot of new data coming out from there. But I think that's always going to be a challenge, and we're always going to have a backlog. But yeah, it's a prioritization problem for sure.
That was my question too.
We have more questions.
Thank you, Mallory, for the overview. Before I ask my question, am I correct in understanding there are plans to include pan-genomes as part of Ensembl?
Yes.
Okay, so if, to that effect, what are the plans? Are you going to construct the pan-genomes from the existing genomes in Ensembl? Or are you going to accept community submissions? Or there are upstream sources from where you're going to pick them from?
Great question. It's more towards the latter. So we're basically partnering and collaborating with the groups that are really doing the research and we're not generating the genome sequences ourselves, but we're collaborating, for example, with the Human Pangenome Reference Consortium. They're generating, I think we have about 90 some haplotype genomes from that project. The visualization of those resulting pangenomes is also a challenge. So we definitely have an eye towards that, but it's first sort of getting all of those references in collaborations with groups, and potentially there's ways to have self-submission in the future, but we're starting sort of basic for now.
We have time for one more question.
You mentioned cost in your slide about when talking about I think the rapid release, and I was wondering how do you decide what to prioritize? Is it like,
Do you want to come to our planning meetings and help us?
do you take community feedback?
100%.
Or is it the steering committee that decides?
Yeah, so we have the standard process. We have a scientific advisory board that meets annually. We engage very closely with the community. So my particular community is more on the human side, but there are 6 PIs in Ensembl, and all of us sort of have our own communities that we engage with. So we have someone who's involved with plant communities, microbial communities. So it's really a challenge to prioritize, but I think we have enough people in the different spaces, and then we come together and kind of discuss, the staffing costs, infrastructure costs, these kinds of things. So it's a group effort for sure.
Thank you very much, Mallory. Thank you all for your questions.